Awesome. So I'm going to make a start, and I've managed to lose everybody by sharing my screen. So let me sort that one out. So thank you very much for joining for our Bioscience Lite session this evening. We're going to be talking about evolution and proteins, and we've got sort of two guests in this space this evening that I'll introduce as we go. So before we start, I'll do sort of the housekeeping bit. So many of you have seen this slide before, but all of our events are aimed to help everyone explore the science of genomics, what it means for our lives, and obviously in this context, what it means for sort of education and learning as well. Me and Fran this evening will be moderating the space to make sure it's safe and inclusive for all, but we do... Sounds like we've got some extra guests on the line as well. If you can just mute yourself whilst we... Awesome. So what are we up to this evening? If my slides want to work. Oh, sorry, my slides had a minute and now we've skipped through the whole presentation. That's a great start, isn't it? So if you've not used Zoom before, if you've managed to avoid it over the last 2 years, you can unmute and chat to us this evening, and you can ask questions as we go. If you'd rather type a question, you can use the chat box for that. We do have closed captions available this evening. If you click the little CC box, they should appear on your screen. You can also move them around the screen so that they're not in the way. And if you do want to ask a question sort of live, you can either raise your physical hand or your little Zoom hand instead. So as I said, myself and Fran are here to sort of host and moderate this evening, and our guest speaker, we have Zachary Arden, who is a postdoc at Wellcome Sanger, and we've also got Bryony Jackson, who's joining us to share a brand new learning resource linked to this topic as well, that will be available for use in the next sort of few weeks. So we're all based here at the Wellcome Genome campus, just south of Cambridge, for those of you that haven't come across us before. And we're a world-leading genomics research institute made up of Wellcome Sanger, which is where Zachary's based, and Emble EBI, which is where Bryony is joining us from. Just before I hand over to Zachary then, we run an educational resource website called Your Genome, and we've been publishing lots of new content there over the last couple of months. This last batch on RNA and bioinformatics dropped a couple of weeks ago, and we've got some new content on phylogeny and evolution linked to Zachary's talk this evening that will be coming out over the next month or so as well. We're also really excited to share that we have a new FutureLearn course coming soon. This slide's pretty blank because we are still developing it, but it will be available for free for teachers and educators to do sometime over spring. So we're really excited about that, and we'll keep you updated on that as well. How this evening is going to go then, I'm going to pass over to Zachary soon, and he's going to talk to us about protein and evolution and his work. We'll then have time for a Q&A with Zachary, but as I said, if you have questions as we go, just chip in the chat box or via microphone as well. After that session, we'll let Zachary go, and I'll pass over to Bryony. He's going to show us the new resource, Our Animal DNA, all around evolution and phylogeny with a bioinformatics twist on it as well. This session is being recorded and it will be available on YouTube and our online learning portal in the next few days. So if you have to dash off early or miss any of it, don't worry, you won't miss out completely. And the title is not CRISPR, it's proteins and evolution. Apologies for that. So I will stop sharing my slides and I will hand over to Zachary.
Great, thanks very much. I'll just share my slides. Okay, so I'll be talking about protein evolution, particularly how evolution makes new proteins in the first place. And hopefully some of you will have questions. This is quite a large topic, and so covers quite a few different areas of molecular evolution. I'm a postdoc at the Wellcome Sanger Institute in Nick Thompson's group. His group mostly focuses on infectious diseases, bacterial diseases, and I work on a subset of that, the proteins that are found in particular pathogens that are not found in their relatives, for instance. and how the processes by which these proteins originated. Okay, so this was, I'm not sure if anyone saw the previous talk or know the context. This was a talk that I gave a couple of weeks ago for school students, and I'll present it again. And if you have questions, please feel free to interrupt, and we can discuss any of these things. So first question, genomes, genes, and proteins, why do they matter? And there's lots of answers you could give to this, but one reason is proteins are the main part of the molecular basis of life, and they're encoded by genes, and together the set of genes is roughly the genome. Proteins are key for understanding function, disease, and evolution at the molecular level. And life, of course, is very diverse, all these different organisms and far more. If we really want to understand all these things at a fundamental level, then we'll want to understand proteins. This is the overview of the talk. The first half is background, research which is reasonably well known, perhaps. So firstly, just a quick introduction. What are genomes, genes, and proteins? What is evolution from a molecular perspective? Then some examples of protein coding genes, and then the standard picture of how people have thought that proteins evolve. And then the second part is more recent discoveries, which kind of changed the picture of protein evolution that the molecular evolution community has had in the last few years. So, of course, a gene is basically the DNA instructions for a protein. There are, of course, other kinds of genes. It's debated how we might define a gene, but this is, in the simplest case, is a gene is just the DNA instructions for a protein. Protein is the string of amino acids encoded by a gene. For instance, the now famous spike protein from SARS-CoV-2, which we'll come back to. And genome is the full collection of all the genetic material in a cell or a virus, including the genes. For instance, the genome of SARS-CoV-2, which is about 30,000 nucleotides long. What is evolution? There's zooming out, we can take a big picture perspective. It's the processes from which life shifted from something like this, single cells, approximately 3 billion years ago, to the huge diversity of life that we see on Earth today. So all those different biological processes which are involved in this, in the mechanisms of descent, that's evolution, a very large scale thing occurring over billions of years. But there's also a small scale of evolution. This is my favorite illustration of that, which is this animation of what's called the mega plate experiment from the Kishney lab at Harvard. MEGA is an acronym that I think stands for Microbial Evolutionary Growth Arena. And what's happening here is that this is a very large petri dish, effectively. It's a couple of meters long. And at either end of the petri dish, the white regions indicates where bacteria are able to grow. The middle, when it's dark, means the bacteria are not growing there. There's some dye in there. And when bacteria grow, they metabolize the dye, I believe, and that changes the color. And they're initially not able to grow in the middle because there's antibiotic chemicals which are preventing bacterial growth. In order for the bacteria to be able to grow, they have to evolve. They have to change genetically in some aspect so that the bacteria are able to survive in the antibiotics. And if you look carefully, you see this kind of stripes on the plate, and these stripes each have an increasing concentration of antibiotic as you move towards the center. So in order to continue to grow past each stripe, the bacteria have to continue to evolve. And this is a sped up video of what occurs over the course of a couple of days or so of evolution. So we really can see evolution happening before our eyes in this kind of experiment. This is an example of what's called experimental evolution. And if anyone has any questions, feel free to call out. What's really interesting for me is that not only can we see evolution before our eyes in this case, but we can go back and work out what's happening at the molecular level. So we can find the genomes of the bacteria at the different stages, and we can see the genetic changes that occurred in order to, and some of these would be adaptive changes that actually facilitated the growth in the higher and higher concentrations of antibiotics. So this is one example of, which I think is a nice example of how we can understand the molecular level of evolution. Of course, the spike protein in SARS-CoV-2 is now famous around the outside of the SARS-CoV-2 virus. And it forms, it's constituted by a trimer of these proteins, which together form the spike complex. This is maybe a more impressive example, which is also fairly famous, which is the bacterioflagellum. This is basically a machine which various different kinds of bacteria use to swim through liquid media. And the next slide is an animation of the core components of the machine. So this is relatively recently discovered through cryo electron microscopy, this complex structure. And this just shows basically how the central units of this molecular machine spin. They actually spin at more than 10,000 RPM, and there's a long tail attached to this machine, which allows the bacteria to swim through their media. There's various different kinds of flagella which are evolutionarily related to each other. Okay, so then seeing these things might raise the question of how do these things evolve? And there's kind of a standard picture of protein evolution, which people have had until fairly recently, which is that proteins basically always evolve in the standard way, which there was an ancestor gene here in purple, and then maybe this gene got copied at some point in the past. If that did happen, then you'd have two copies of the gene, which are just these rectangles side by side. And the standard picture is that they just diverge over time. Maybe the original one retains its original function and it doesn't change much. I'm just shown with a relatively minor change in color here. Whereas the second copy, because it's not functionally constrained, because the original copy is already doing the original function, the second copy is able to diverge more significantly, and then you get a different sequence, a very different sequence here. And if you looked at these two sequences, at least superficially, at the sequence level, you might not realize that these two sequences were actually related through a common ancestor. And so this, this approach is called various related terms, such as duplication and divergence. And this was the standard model for protein evolution, which people had. This is not just a theory, it's known to have happened in many cases, including these two genes, which are in SARS-CoV-2. So these two accessory genes, ORF7A and ORF8, both actually had a common ancestor, which was basically like ORF7A. So the ancestral copy of ORF7A got copied, and one of those copies resulted in the ORF7A that we see in SARS-CoV-2, and the other one resulted in this quite different protein, ORF8. And there's lots of other examples like this. Okay, so this is another illustration of it. You can picture it on a tree. In the first place, you have an ancestral protein, and it's diverged over time into these different sequences represented with different colors, which are found in modern day organisms or viruses. But you can also imagine that maybe at some point in the tree there was a duplication, and then which is illustrated by these proteins here, and then each of these two copies could potentially diverge over time. Yeah, so this is the standard model for what we used to think about how proteins evolve. It has been called in some papers the Big Bang model, and that's because if you kind of follow the logic back, then it implies that nearly all proteins can be traced back through their ancestors to near the origin of life. So the origin of life, there was somehow this initial relative diversity of proteins, and since then, most proteins originated just through copying, just through duplication and divergence over time. And this was basically the standard model that was found in the literature from the 1970s through the 1990s and a bit beyond. And there were good reasons to think this, which we could talk about if people have questions later. And it is true for many key proteins, which were studied in the early years of molecular biology, but it's not true for all proteins, as I'll explain. Just wait for the slides to load. Okay, so a good example of this is the various proteins involved in ATP synthase. These two structures look basically the same. It's basically the same molecular machine, but it turns out that one of these is found in the mitochondria of cows. This one is found in E. coli bacteria. So this is an illustration of that, and that the proteins really haven't changed that much over billions of years. So this is true for some proteins, whereas other proteins have diverged significantly over billions of years. And so as we got more data, the picture changed. Now, the next part of the presentation is the new kind of findings that have been found in the last 20 years, basically due to genomics, as I'll explain. So a lot of this newer material is really new and still being explored. Science is constantly in development, and some of the most interesting stuff in science is found in the parts which are still being worked out. So a lot of these things are still kind of open research questions. Why did the picture change? The answer is basically genomics. We suddenly, since 20 years ago and the last decade in particular, we've had access to a lot more sequence data, a lot more genes and genomes, and we've got a much better sense of kind of the global picture of a protein space, of how proteins have evolved over time. So for instance, what really started this genomics revolution was probably the Human Genome Project, a third of which was conducted at the Wellcome Sanger Institute. This was completed in about 2003, and it opened the doors to sequencing thousands of different organisms. A recent update on this is called the Darwin Tree of Life Project. This is just one example of a very large genome sequencing project. The aim of this project is to sequence all the animals and plants in the United Kingdom. And again, the Wellcome Sanger Institute is very much involved in this. Okay, so what we found when we had all these new genome sequences is that many genes are relatively young. So this is illustrated nicely here. So imagine that this is part of the genome of Luca, the last universal common ancestor. What's depicted here is that it has relatively few genes. So it just has these, whereas extant genomes have a lot more genes, illustrated here with the different colors. And what we see is these different groups on the tree have different colors represented. So for instance, this yellow gene is only found in this group B. What's that's showing is that many genes are only found in a small taxonomic group. For instance, maybe they're only found in one genus or one family or one species. This was quite surprising for a lot of people who had kind of assumed the previous model of duplication and divergence. And this whole area is called the study of orphan genes or taxonomically restricted genes or gene origins in general. And this is really a rapidly growing field in the last few years. Yeah, so what was discovered is that a lot of these genes are young, so they don't have common ancestors, and that raises the question, okay, where did they come from? So that raises the question, what's the mechanism by which these genes arose? Potentially, you might conceivably be able to explain it through kind of rapid divergence, which is shown here, that you have an ancestral gene, maybe they get copied or they just diverge. And then the endpoints that we see today are so diverse that we can't recognize that they did actually have a common ancestor. So this does explain a certain proportion of genes that look like they're young, but not all of them by any extent. The alternative process is something called de novo gene emergence, de novo Latin from nothing. And it's called that because these genes actually, they're protein coding, but they arose from non-coding sequence. So there was some ancestral sequence, which was not a gene. It was nucleotides, but it wasn't involved in a gene. it mutated to the point where it became, for instance, an open reading frame that got translated and became protein coding through various different processes that might have happened. This would be an example of de novo gene origins. This is a really nice kind of review of it from a couple of years ago. Okay, so this was a poll question which we asked. I don't know if maybe Em wants to say something here about.
This is one of the questions we asked to the students in the main student talk. So if you'd like to have a crack at this question, feel free to put any guesses in the chat box. You may well know the answer to the top one, but I think the second part of it really brought up some interesting ideas from the students. I think a lot of them, John's got it straight. John's gone for 2%. Zachary, you'll confirm whether that's correct, because I believe it's 2%, or was it 0.2%? It's 2%.
Yeah, 2% is right.
I think we had a lot of great answers from students, sort of knowing. I think we would particularly see a good shift towards calling it non-coding DNA rather than the dreaded junk DNA. And we had some great ideas about what DNA does apart from encoding proteins. So it was really nice to see that a lot of students knew about some regulatory aspects of this DNA as well.
Right, yeah, so in asking this question, we were interested in just making clear, I guess, the distinction between coding and non-coding, and realizing that in some organisms anyway, only a tiny proportion of the genome is actually protein coding. In bacteria, it's more like 80 to 90% of the DNA is protein coding, and it varies widely across organisms. So what is non-coding DNA? Some regions of non-coding DNA have sometimes been called junk DNA. This was just a term that a lot of people get confused about, but now it's now known there are many different things that non-coding DNA can do. It's basically always been known since the origin of molecular genetics that there's some things that non-coding DNA can do, but we've increasingly found more and more functions for a certain proportion of this. These are just some examples. And in general, insofar as they have function, it tends to be something to do with regulating the expression of protein coding genes. There's lots of different kinds of RNAs, most of which I don't really know much about. This is a controversial topic. We don't actually know what percentage of the human genome is functional. It's tricky how we define function. And so it will depend what we mean by function. If any of you have questions about this, we're happy to discuss later. It's also debated in the literature whether junk DNA is a useful term at all. It's quite a complex debate, but the main thing to take away is that it can't be used to describe all non-coding DNA. So there's a reasonable portion of non-coding DNA which is functional, and whether you should call the rest junk or not is an active debate. There's a nice section on this on the Wikipedia page on non-coding DNA. Okay, so how can a gene be born? How can you get a new protein coding gene from something which didn't used to encode proteins? One of the best studied examples of this is a protein which is found in fish which live in very cold water. This is an antifreeze protein, and it's been shown that the ancestral sequence looks something like this. They were non-coding, and then over time, and we can kind of see this if we trace the phylogeny, if we see that these groups here lack the coding sequence, and only a restricted group actually have, say here, have some kind of coding sequence. And through ancestral sequence reconstruction, we can kind of work our way back and infer what the ancestors of these groups as we go wider and wider in the taxonomy, what that sequence must have looked like. So this protein coding sequence with the various genetic elements is restricted to a small taxonomic group. And that's the main point. Just this case in particular has been studied quite closely, and we've been able to work out from which genetic region the protein evolved. So the protein coding sequence evolved, and that genetic region in the ancestors wasn't protein coding. There's numerous examples of this in the literature. This is just one of the better studied ones. Another main thing to think about non-coding DNA is it's not all the same. And this is the kind of thing that my research is focused on. So an open reading frame on the ORF is any part of the DNA which is between a start and a stop codon, any region of the genome which is uninterrupted by stop codons and the reading frame which we're looking at. There's many of these open reading frames, many hundreds of thousands of these that are not transcribed or translated and don't encode proteins. And this is an important distinction that a lot of people get confused. An open reading frame is not the same thing as a gene. Different open reading frames have different properties. For instance, codon usage, which codons are more frequent or frequently used or not frequently used, GC content, the nucleotide content, the probability that they will form a folded protein if they are translated, and various other sequence properties. And this is what I mostly study in bacteria. These are illustrations of the genetic code that I'll come to soon. Many open reading frames are overlapping other genes. These are generally ignored, but they have some interesting properties. And some of these properties, if you dig into it, are actually given by the structure of the standard genetic code. This is an example. So you're probably all very familiar with the genetic code. You might not be so familiar with the fact the code has an arrangement, which means that the chemical properties of the amino acids are reflected in the arrangement of the code. So this top picture shows the structure of the genetic code, and the colors show the chemical properties. So they're classed into, I think, five biochemical classes, the amino acids. And it turns out that chemically similar amino acids are clustered together in the space of the nucleotide space of the genetic code. That means that a mutation at the nucleotide level is unlikely to have a large effect at the amino acid level. If it does change the amino acid, it'll change it typically to a relatively similar amino acid. So that's an interesting property of the code that's been studied quite a bit. There's other properties of the code that also affect what happens in overlapping reading frames. So if you shift the reading frame, if you shift the codon you're reading, the properties of those codons are determined by the genetic code. An example of this is that the codons are also clustered so that the amino acids that are similar, the hydrophobicity, are located in a similar region of the code. If you change the reading frame from the sense to the antisense, so for instance, if you change from the triplet UUU, which is here, if you change that to the opposite triplet, this would be AAA, which would be somewhere there. Sorry, so A, and then the fourth, there, yeah, so it'll be here. This will have the opposite hydrophobicity. So you go from hydrophobic to a hydrophilic amino acid. So these kinds of changes from sense to antisense by kind of a strange feature of the code means that you tend to go from a hydrophobic to a hydrophilic amino acid. Yeah, so the properties, the main thing to take away is that the properties of the overlapping genes are also determined by the structure of the code. That means that some overlapping reading frames are more interesting chemically than others. They have chemical properties which are more similar to protein coding genes than other open reading frames. So not all open reading frames are the same. That was quite a technical thing, and if you want to dig into it more, you can look into some of the papers in this area of overlapping genes. This is another example, which I'll just kind of skip this and say the main thing. These are some scores that have been given to open reading frames. These are the open reading frames that are actually annotated as genes. And these are all the open reading frames that are not annotated as genes. And there's a lot of these that actually have a reasonably high score. In terms of the score basically says how gene-like these open reading frames are. So in other words, there's not a sharp dichotomy between open reading frames that are genes and open reading frames that are not genes. There's actually this whole region in the middle, which you could call the shadow zone. So some open reading frames are more interesting than others. They have more interesting properties that make them more likely to become genes in future. Some examples of protein origin from SARS-CoV-2, the virus that causes COVID-19. A couple of examples are here. So these two genes here, ORF3A and M, are actually related, again, through this mechanism of duplication and divergence. So the ORF3A protein actually had an ancestor, which was basically the M protein, the membrane protein. And this got copied, and then you now in the SARS-CoV-2 genome, we have two copies, which are two separate genes, which are quite different from each other. And similarly, the ones I mentioned before, ORF7A and ORF8. So these mechanisms of protein evolution, where you get, in some sense, a new gene, either through duplication divergence or from non-coding sequence, are happening all the time and are influencing the evolution even of SARS-CoV-2. I'll skip this slide. This is just an illustration of the different reading frames. In a genome sequence, you have 6 reading frames because you have 3 on the plus strand and 3 on the minus strand, and you can show the genes that are present and some of the sequence properties along the genome. Okay, so some questions to think about from the things I've mentioned. Why are proteins important? How do you personally think about evolution? And I guess in light of this talk, you can think about it maybe more in terms of a molecular process, as well as in terms of changes in phenotype. What's the link between evolution and proteins? And why does the evolution of proteins matter for understanding proteins themselves? Also, maybe are there some things about evolution that we may never understand, given that we're always kind of learning more about evolution? Are we eventually going to unpack the full story of molecular evolution, or is it inherently some things which are too complicated or too far in the past that we'll never quite fully have a detailed mechanistic account of? So with that, I'm very happy to take any questions.
Awesome, thanks so much, Zachary. Yeah, I always enjoy hearing your talk. So if anyone's got any questions for Zachary, then either unmute and ask away, or pop them in the chat box. We've got a little while to ask some questions. Ian, I can see you're off mute. Are you got a question for Zachary?
I've got one too. How easy is it to discover a function for non-coding DNA?
Yeah, that's a great question. So I guess it depends for what organism we're talking about. Like, at least with bacteria, we can do a whole bunch of experiments, which, yeah, so in some eukaryotes, that's going to be more difficult. But for bacteria, sometimes we can see that there's like an RNA that's being transcribed, and we can maybe see what the RNA is binding to. If we do some study of RNA protein binding, well, there's all kinds of biochemical tests that could be done depending on what kind of genomic region it is. Like, maybe it's a transcription factor binding site, or maybe it's actually an RNA, or there's lots of experiments, and some kinds of RNAs are going to be easier to find a function for than others. And there's a lot of the stuff that we really don't have much idea. We just see that it's been transcribed. We don't know what that's doing, if it's even doing anything functionally.
Yeah. Okay, thanks. Just to say that I'm an A-level teacher, and the concept of the gene has changed regularly over the years. And, when I'm talking to my students, what is a gene? What would be your answer to an A-level student now who said, what is a gene?
Yeah, I don't have a great sense of where A-level students are at. But to start with, so I guess I would say that there's kind of different levels of definition. And the easiest definition is that it's a region of DNA which is encoding a protein. But then I'd say, but there's also a wider concept of a gene, which is any region of DNA which is encoding something functional, including the various kinds of RNAs. Yeah, I think that's the best definition that I can give.
Yeah, okay, thank you. And my third question, if I may ask another one,
Sure.
I started teaching 47 years ago, and there were 20 amino acids defined by their R groups. I still teach, there are still 20 amino acids, common amino acids, defined by their R groups. Could we get an evolution of a new amino acid with a new R group, which could change protein structures?
Yeah, so there's another, I really don't know much about this, but like there's another amino acid, selenocysteine, I think, which is sometimes incorporated into proteins. And I should know more about this, but I really don't know how that works. I think there's other cases of this where there's some other amino acid that occasionally gets incorporated. There's also all kinds of like post-translational modifications. So I translate the protein, you have the amino acids, but then other chemical groups get added on top. So that's kind of an extra nuance that gets added. And even there's also modifications at the RNA level as well. And there's, I think, hundreds of these kinds of modifications. So there's, even within the kind of standard 4 bases and 20 amino acids, there's a lot of extra kind of modification that happens on top of that.
Yeah.
Yeah.
So there's lots of redundancy in the system. There are 64 triplets, there are 20 amino acids, so we could happily transfer a triplet to a brand new amino acid.
Right. So yeah, one of the interesting consequences of that redundancy is the stuff about overlapping genes that I tried to explain very badly. I should have had more diagrams of that. But the fact you have this redundancy allows you to encode extra additional information within a genetic sequence, including overlapping genes in some cases, but also regulatory motifs are kind of buried within the gene. So the fact that the fact that you can encode one amino acid with potentially 3 or more different codons means there's quite a bit of kind of coding flexibility in exactly which nucleotide sequences you use in order to encode the amino acids. So that redundancy is not actually redundant in the sense of like useless. It's actually used to allow, like, also codon usage is really important, and it controls the speed of translation. In some cases that controls protein folding. So yeah, it might look redundant on the surface, but it's actually, a lot of it is actually useful.
Yeah, okay, thank you.
Awesome, thanks for your questions, Ian. And then just to piggyback off of your first question, Ian, we've done a previous session around sort of CRISPR and doing knockout experiments to understand function for disease purposes. And our speaker's also written a really great fact page that I'll stick in the chat box that goes through how CRISPR could basically knock out a gene at a time to help us understand exactly what's happening in the disease state. So that might link a little bit to your
lovely.
Your first
Thank
one.
you.
Zach, we've got a few questions in the chat box that I'll read out. I got one from John asking, why is the number of genes relatively similar across all life? So quite a big question, but I wonder if you've got any insight on that one.
Yeah, that's a really good question. And the standard answer is that in more complex eukaryotes, they're regulated differently. And that doesn't explain why it's the same, but it just explains how it's the same in the sense that it's able to be the same, because yet still produce more complexity because the regulation is different. So in humans, you have hundreds of different cell types. I think there's more than 500. I think there's more than 800 different cell types in the human body, and these have different like transcriptional and translational regulation. So it's because of the in short, you could say it's because of the non-coding DNA that you're able to produce more complexity with the same number of genes. I think it can be helpful to think of genes as like Lego blocks. You can arrange them in lots of different ways. and the stuff that's controlling that arrangement is a lot of the non-coding DNA.
Awesome. And then 2 questions from Haley around open reading frame. So the first was, when you say open reading frame, do you mean that those sections of DNA aren't as tightly coiled around histones, but they aren't genes because they don't have a promoter region, so aren't transcribed? Quite a long one, so it's in the chat box if you want to read through that one again.
Yeah. So an open reading frame is just any stretch of nucleotides that's between a start codon and a stop codon, or some people define it as any stretch between 2 stop codons in the same reading frame, so in the same reading in the same set of triplets. So that doesn't tell you anything about whether it's transcribed, whether it's translated, or whether it's with the epigenetic control of it, or whether it's how tightly it is bound by histones or its methylation or anything like that. It's just a region that is an open reading frame because it's not closed by a stop codon. And some of these are subsequently transcribed because they have some kind of promoter region. And then some of these that are transcribed are also additionally translated. And then, so if you have transcription and translation, then you produce a protein of some kind, and then I would call that kind of a gene in the classical sense.
Helps if I mute myself. And then as a follow-up question, she asked, do these open reading frames change into genes by mutations in the code or something else? I know that we had a similar question come up in the student talk as well around that.
Yeah, so that process of getting from an open reading frame to a gene is kind of unclear because there's a lot of open reading frames that aren't genes, but there's kind of classical motifs that are associated with being a gene, like a promoter region. In bacteria for translation, you also have the Shine-Dalgarno region, which is not always there, but it's quite often there. So there's these kind of genetic signals that seem to tell the cell's machinery that this section of the genome is not just another bit of DNA, but is actually meant to be transcribed, and then it's also meant to be translated. Yeah, I think that's the best answer I can give.
Awesome, thank you. We've probably got time for a few more questions if anyone has any more. I'm just going to check in the chat box if we had any others.
Can I ask one more?
Go for it, Ian.
Yeah, several years ago, I had Sir John Walker into college, who got the Nobel Prize for his work on mitochondria ATP synthase. And he said that the genetic code was slightly different in mitochondria to, the genetic code that you posted earlier. Is that correct? Yeah. I'm not saying John's wrong.
Yeah, he knows what he's talking about. Yes, that's right. Yeah, so there are some different genetic codes, but they're all. And I should know the details of these. There's I think at NCBI there's at least 11 codon tables. I think there's a few more, but if you look at them carefully, you see they're all very minor changes. And because they're highly taxonomically restricted, you can infer reasonably that they're all derived actually from the standard genetic code. So they all just have a couple of codon changes, like one codon that used to encode an amino acid now encodes a stop codon. That's the most common change, as I understand. And not only are they highly taxonomically restricted, they're also usually in organisms or in organelles that have a really small proteome, so they don't encode many proteins. Yeah. So that means that the change, the switch in the code didn't have that drastic an effect. Because this is a kind of classic question, how could you possibly change the code, given that whenever you change the code, you change all the messages encoded by the code? And part of the answer is, well, you can change it if the proteome is very small, so there's not many messages that are changed. And that's the cases where we see a change in the code is usually that situation, including the mitochondria, which doesn't encode many proteins. So changing the code is kind of possible.
Yeah, thank you.
Awesome. Again, yeah, if anyone's got any last questions, we can take one or two more. Otherwise, we will shortly move on to Bryony's talk about our new activity. So yeah, I'm just having a look through the questions we had from students, and a lot of them were similar to what came up this evening. One that I think was really interesting that maybe I'll ask you now, Zachary, was the question around at what point do you classify a new bacteria as a new species? Like at what point in the sort of mutation set? So I wonder if you could briefly talk about that as well.
Yeah, I can't remember the answer I gave last time, but I probably said something like it's really subjective, because at least with sexually reproducing eukaryotes, there's kind of the classic definition, which is like an interbreeding population or something like that. You can define that as a species. In bacteria, there's these kind of standard thresholds. I think it's something like a 97% similarity for a particular gene. I think it's the ribosomal RNA. Is classically used as kind of the threshold for a new species, and then maybe it's 95% is for a new genus or something like that. There's kind of approximate thresholds that are used. Yeah, it's somewhat subjective, but at the same time, these thresholds do correlate with other biological features. So organisms that are part of the same species are much more likely to share genes. Well, yeah, they have other genetic features in common as well. So it's not just an arbitrary threshold based on one gene. These things do somewhat correlate, at least. Yeah, but with bacteria, it's hard to draw the precise thresholds because they're not sexually reproducing. Yeah.
Awesome. Yeah, I think that was more or less what we covered last time as well. In that case, Zachary, thank you very much for joining us. I will let you wrap up and give you back a little bit of your evening. Yeah, thank you so much for joining us this evening. And if anyone thinks of more questions afterwards, feel free to email us and we can put you in touch with Zachary to sort of chat further. But I will now pass over to Bryony, who will introduce herself and then share our new activity linked to phylogeny and evolution. So yeah, over to you, Bryony.
Thanks, Em. Can everyone hear me okay? Perfect.
Yeah.
And the screen sharing works smoothly, that's exciting. So yeah, good evening everyone, and thanks Zachary, that was a really interesting talk. I'm Bryony, I work for EMBL EBI, so a lovely acronym, science love an acronym. So we are the European Bioinformatics Institute. We're part of EMBL, which is the European Molecular Biology Laboratory. It's a European-based organisation. We've got sites across Europe. EBI is the UK-based site, and while the other sites do a lot of traditional wet lab work and focus on basic biology, we focus purely on computing science and bioinformatics. So our scientists are all computer-based, which was useful in the pandemic when everyone had to work from home. They come from a range of backgrounds, so programming, coding, software, and the kind of traditional life sciences backgrounds as well. And I think that's kind of a really interesting story to tell students, that the kind of different roles that are required in this sector of the science world. And we're based on the Wellcome Genome campus, so we're neighbours to Emma and Fran, and we have a number of databases which are free open access databases for anyone to use, but obviously they're a bit more accessible to the science community. The databases hold data from wet lab experiments, and the one that we'll be kind of introducing today and the one that's covered in the activity is Ensemble, which is a genome database. Our scientists kind of curate and annotate the data that's submitted from all around the world, and then it's freely accessible for other scientists to come in and take that data and use it without having to do those experiments again. So it speeds up the kind of scientific discovery process. So I will give a quick overview of the activity. So it's called Our Animal DNA. It's part of Geometry of Life. So Ensemble has a rapid release portal which puts out the genome sequences that are coming through from the Geometry of Life work, with not as much information as the traditional releases. They just have data set, so a gene set and functionality only. and more data will be added later on, but it's kind of a quick access route for people working on that project. So we wanted to give students a kind of insight into geometry of life, some of the bioinformatics elements involved in the project, and also a kind of having a go at using some tools in a real world scientific context. So we've aimed it at A-level students, so year 12, 13, S5, S6. It's a downloadable resource, so it's a video workshop which is led by one of our scientists, Emily Perry, and then there's some computer-based activities using 2 of our bioinformatics tools, which are all web browser based. Don't think you have to download anything to go with it, no Flash or anything like that to download. It takes about an hour, and you could split that into 2 sessions or have it as 1 hour activity. And there's expansion potential in that students, once they kind of have the skills and having been introduced to the tools, they can explore as much or as little as they like. It's free for participating schools and colleges. It's going to be available to download from your genome. In the interim, I can also kind of send the Google link to any interested teachers as well. And all that's needed is a computer per student with internet access. And our aim for this is really to excite students about the kind of current activities in genomics and DNA research, open their eyes to the field of bioinformatics and how it can be applied in this setting, share those tools, those bioinformatics tools that they can go away and explore, and then also introduce those career and study options, particularly in the field of bioinformatics, which are really increasing, and where life sciences and computing meets, and the potential there for careers and study. So okay, this is where technical pictures will occur. So I wanted to give you a quick overview of bits of the video that Emily's recorded to accompany the activities. So the activities are worksheet based, but the video from Emily supports the introduction of the topic and supports you to support your students to work through the activities. She does a lovely bit about her career path and how she got to it. So I'm going to attempt to play that for you now.
Can't hear the sound, Bryony.
I think you need to click share sound on share screen.
Ah, okay. Oh, this is exciting and new.
Might need to stop sharing in order to do it though.
Oh, okay. Sorry, this is going to stop it being smooth. Right, stop share.
And then as you reshare, there should be a button bottom left that says share sound.
Oh yeah, share sound. Awesome. Yay.
Magic of Zoom.
All right. Let's pretend that didn't happen. This is a lovely clip from the video.
Before we get stuck into the science, I'm going to tell you a little about me, how I ended up in the job I do, and what working for a large science organisation is like. I went to an ordinary comprehensive school in Lancashire. I did sciences for GCSE and A-level, but I also did a bit of drama. Then I went to the University of Edinburgh, where I studied genetics. Then I stayed on and did a PhD. A PhD is another degree that you do after your undergraduate degree. During a PhD, you work on a long-term research project. Mine took 3 years, then write a thesis at the end. For science PhDs, you often do these in a lab. It turns out that I'm terrible at lab work. I am clumsy and I am impatient, which are not good in a lab.
So that's just a bit of a clip. So it's nearly ready, the video to share. We're just kind of building in some animations. So throughout the video, Emily talks through, has some animations to support the concepts and topics that she's talking about. So you can see the screenshot in the bottom there, looking at comparing genetic sequences. She also has built in some kind of interactive options. So points where you can pause and discuss things with your class, so it doesn't become too passive and too kind of screen watchy. So asking to discuss bioinformatics, she asked about the potential applications for bioinformatics and what it can kind of, how it can help speed up science. And so there's a few points throughout the video for that to happen. And then she'll introduce you to using BLAST and Clustal. The worksheets have all the information that you need. There's a worksheet for teachers to use, and then the one that the students will follow to lead you through the whole activity. So yeah, this is an example of the animation, which hopefully will play. So this is talking about alignment. It's a bit bright at the moment. This is one of the ones we're adapting to make it a little bit less eye melty, but it just helps support what she's introducing to students. So the topics that she'll kind of bring in are evolution and talk about morphology, and then orthologs and how using those various bioinformatics tools allows a comparison and discovering functions within the genes and comparing new species, and especially for the species that are being sequenced through Darwin Tree of Life. They've never been sequenced before, so there's lots of potential new information to discover there, but also a lot of data coming out at once. So how we can really use these tools to maximize what we can find out and in a kind of short space of time. And as I say, there's potential once the students have those tools and those skills, they can go on and discover whatever they like about all different species that are coming out. And as the Darwin Tree of Life project goes on, there'll be more and more sequences released on that portal, and they'll be able to kind of keep dipping back in, seeing what's there, seeing what other comparisons they can draw. Oh gosh, no, it's playing again. How do I make it stop? There we go. Yay. So yeah, that's just a brief overview. As said, it will be ready in the next few weeks. Then it will be available on your genome to download. And if you have any questions about it in the meantime, or you want to register your interest, we can let you know when it's live. And if you have any problems working through the activity or anything's not working, then do get in touch with me. I will go and ask a scientist and get them to help. If anyone's got any questions now, please feel free to ask them.
Can I ask you a question, Bryony?
Do.
Can students register and engage with this on their own at home? One of the problems we always face in schools and colleges is we have such a packed curriculum that shoehorning things in class is really difficult. But if we've got keen students, can they do it off their own bat?
Yeah, I would be confident that they could. I don't know, Emma, Fran, what you think. I think Emily does kind of, she leads you through it really well. It's the only bit that they would miss is that opportunity for discussion. Sure. With you or with their classmates, but they could think about the answers on their own, and then Emily does give responses to that as well. So yeah, I don't see why a motivated student should be fine. Also, it could be quite interesting for any kind of students that have an interest in computing as well, that it might be off curriculum a bit for them, but it might be something that's interesting to them.
Yeah, I definitely say it's suitable for particularly sort of enthusiastic students that want to sort of tackle this in their own time. Yeah, as Bryony said, the video, she definitely sort of, presumes that you're watching it as a class, but yeah, we've scripted it in a way to make sure it's got all the background information they need, so they can definitely go off and do it. It's also sort of a two-part activity. So again, one option might be that you do a small amount in class and then they go off and do the second activity at home. So I'm quite flexible to be split between sort of class and home learning. Yeah.
Okay, thank you.
Yeah, and once it's on your genome, in theory, because it's open website, anyone could download it, and the resources, because everything they need will be on there. So yeah, I think that's absolutely fine.
Yeah, and we can promote it, recommend it to our students if they're interested.
That would be great. Yeah, and actually for the databases, if they have any problems using their tools, there are kind of help options and contact us options there, because scientists have problems using them sometimes if it's new to them. So if they got really stuck and they kind of hadn't had a response from me because I'm part-time, then they could always go through that help section on the browser as well.
Yeah. So I'll probably be just to say, because we're just tidying up the resources, so it'll probably be about a month before it's fully available on your genome. But if you want it before then, we could probably send you the files separately as via email or via Google Drive. So yeah, just email us with that as well. And also just a cheeky ad is that if you're interested in the wider Darwin Tree of Life project, there is we've collaborated with I'm a Scientist, so there are like monthly chats as well, which I can pop in the chat there. So if your students are interested in this project, from the concept of sequencing all the life on Earth, there is this they can actually sort of engage. It's not video chat, it's text chat, but they can chat online. I think there's one every month, so that might be a nice thing for students to engage with as well.
There's a question in the chat box from Gemma saying, is this aimed more at biology students or would it be suitable for cross-curricular collaboration projects with computing learners? I'll let you lead on that, Bryony, but I think I know the answer.
That's a fab question, and yes, it's for everyone. So although it was designed in the context of the kind of life sciences, we're really keen that it could be used by computing students as well. Again, if there's any extra support needed for that or more kind of additional worksheets or context, then do let me know. But yeah, I think it's a good kind of a theme topic, but it's a great way to use those computing tools in a slightly different way.
I think it's really nice that this brings the two together as well, because we want to show that actually biology is changing. It is becoming more computational. So actually sort of bringing the two together. So maybe getting computer science students to sort of think biologically and biological students to think computery. That's a technical term, clearly. You know, so that I think it's really good. It's really great to have that.
Yeah, and I think that's the nice thing where Emily talks a bit about her career. She did come through the kind of, she studied genetics, but as I say, we have a lot of staff here that are coders, programmers. They've worked in software development, because not just the handling all the data, but kind of making those kind of user-friendly, front-facing browsers, all that sort of thing requires all those extra computing skills. So there's so much scope for different career opportunities within the sector.
Yeah. Can I say that one of my students a couple of years back could quite easily have gone off to university, chose not to, and took up a degree apprenticeship in bioinformatics at the Sanger. And, you know, I fully endorse those opportunities. University isn't for everybody, and I'm sure he's going to do really well.
Oh, that's fantastic to hear. And I think the more of those sorts of opportunities, I mean, I can speak for myself. I wasn't keen to go to university. I actually waited 2 years to go because I just didn't think it was for me. And if there had been a kind of more workplace-based option, it would definitely have been of interest. So I think they're really important to have that option. And I think these sites that sort of crossover career paths have good opportunities for that.
Yeah.
Well, we might be able to wrap up a little bit early this evening. If anyone does have further questions for Bryony, feel free to put them in the chat box. Bryony, if you just put your email in the chat box as well, that'd be super handy, so that if anyone wants sort of sneak peek access, Bryony might be able to get you that, or just more questions about using the resource, then Bryony is is leading on the brains half of it, so she can.
Just had a thought actually. If you're not already on our It's Learning platform, then email us and we can get you added. But maybe, Bryony, we could put it on the It's Learning platform as well, so people can access it via there before it's on your genome. So if you haven't, so It's Learning is just like a virtual learning environment that we use just to put things up there for teachers to access easily. So if you're not already on our platform, just email us with your email address and we can get you added, and it's very easy to use. So that's education@welcomeconnectingscience.org, and we'll get that set up for you.
Yeah, that would be great. And if anyone, because this is sort of a bit of a new resource for us, isn't it, having that kind of crossover, so if there's anything you think doesn't work well or you particularly like, please let us know. Feedback is always welcome. And as I say, any kind of issues using it, then just ping me an email and I will attempt to help.
Very good. As a question, is there a Padlet for tonight? But I think we didn't have time for a Padlet this week, did we? Because it's been a bit mad.
Yes, we've not got a Padlet. So for those of us that have joined before, we sometimes have a Padlet for sort of sharing ideas, because this week we had Bryony more just sort of sharing a new resource. We've not got a Padlet, but if you've got resources, John, that you'd like to share with us, feel free to let us know. And likewise, once this is uploaded to the It's Learning page, there's sort of forum discussion areas that, again, you can share any thoughts and ideas and resources with each other there. Haley's asks, thanks, the resource sound great. I'm not on the platform, but it sounds great too. Do we need to put anything specific in the subject box? If you can just put Bioscience Lite It's Learning, or words to that effect, we will know what it means. But yeah, thank you for asking before we get very confused about what our emails are about. Awesome. Fab. Well, in that case, I think we will wrap up early this evening, which hopefully is a nice surprise for everyone. Thank you very much for joining us this evening. Our next session will be next month on sequencing malaria. So we've got a student-facing talk at the end of this month, and then we'll do our teacher-facing talk a couple of weeks later, and we'll send out information on our mailing list and on the It's Learning platform as well. So yeah, if you're worried you're going to miss anything, drop us an email and we'll make sure you're on all of the relevant mailing lists. But otherwise, yeah, thank you very much for joining us, and we'll hopefully see you again soon.